Okay, please take a seat. We'll resume the conference. So, welcome everyone after lunch. It is my great pleasure to introduce you Noelia Ferruz. Noelia did a chemistry degree in Zaragoza, and then she did our master's program of bioinformatics, from which I'm very proud. That's where she went so far. With her training, she did her PhD here at the PRBB with Jenny Fabritis on molecular simulations. Then she moved to Boston, to Pfizer, where she did some experience in the industry, and then she went to Germany, to Bayreuth, where she started pursuing her current interest on protein design and protein engineering. And after a very successful postdoc period, she joined the CRG here at the PRB as a principal investigator. And so thanks for accepting the invitation, Noelia. We look forward to the talk.
Thank you. Yeah, thank you very much, and thanks, Robert, for the very kind invitation. It's a pleasure to be here, and even if we come a little bit from maybe we're outsiders of the field, I hope to give you an overview of how we are using large language models or the architecture behind, like transformers, to design proteins that don't currently exist on Earth. So we work, let me see whether this works. There we go. So we work in the field of protein designs because proteins are the most amazing machines we have on Earth. Every little colorful dot that you see here in this figure is a protein, and they are basically the workhorses of the cell. So they carry all the functions that we have in living beings. They allow me now to speak. They are digesting food and coffee that we right now had. They also allow trees to breathe and produce O2 as we are literally sitting here. So they are amazing nanomachines, very efficient, and it will be incredibly powerful to be able to build other proteins with us functionalities as diverse and powerful as these ones. But it has been traditionally very, very hard. So researchers tried for many years to design protein sequences as these ones, but we miserably failed. Originally in the field, we were trying to design proteins by learning physics, because we happen to know a lot of the physics of these systems. So we know, for example, whenever there is a protein, we happen to know the 3D coordinates of the system, so we know how the distance between atoms, and we can, with Coulombic forces and Lennard-Joule potentials, we can compute those contributions. We also know what are the forces of when a residue is in contact with water in the solvent, so we can also characterize those interactions. And we also know a lot of how the protein folds in 3D space, so what are the angles and torsional dihedrals and so on, to model how a protein should be behaving in space. So this is what people did for 2 decades. People were trying to compute very large energy functions made out of physical components, so physical terms, and then this will be represented as a 3D landscape, and then people will try to find the most optimal solution. This will be a given sequence that falls into a 3D scaffold of your choice. Although this was very computationally expensive, it worked quite well with some limitations. So we see here, does this work? Yes. This is the number of protein designs that were reached out for every year. So we see that in the 90s, we were designing globally, we as the community, the entire world, we were designing 5 or so proteins per year. Then in the 2000s, people were introducing better algorithm solutions. So we were able to design maybe 10 to 20 or 50, even a few more proteins per year. And then since the 10s, there was an explosion in the field. So suddenly we had much better computers, and so using this sort of physics-based algorithms, we were able to design many more proteins. What we are seeing, however, is a change in the slope. So since 2020 or so, we've been able to design many more proteins, and not only they are able to fold in space into a structure that we may want, but also these proteins may be able to have the function that we need. So for example, we can use a protein to capture CO2 or to use it as a vaccine. And this change has been produced by the advances that we are seeing in deep learning. So deep learning and artificial intelligence have overall changed the protein design field, and this was reflected in the Nobel Prize last year in 2024. We are seeing a revolution in AI, and I like to categorize that into language, computer vision, and video generation. And these three information channels are what directly impacted protein research as well. And that's because proteins are, without doing anything, they can be very easily represented into these three information theory levels. For example, we can easily represent proteins as FASTA files, so like a concatenation of letters. And that is very, very similar to natural language. So we have an alphabet, which are the amino acids, and those letters combine into secondary structural terms, the same way that the alphabet, the letters that we have in English, they combine to form words. Those words combine to form a sentence that carries a meaning, the same way that those elements combine into a protein domain that may perform a function. And then those domains, they usually assemble into quaternary structures, the same way that then we have sentences combining to form paragraphs and so on in longer text. So the parallel between protein sequences and human language is very, very clear, which is where we see this massive success of using language models literally for protein design and DNA sequences as well. But proteins are also three-dimensional, so they fall into 3D, specific 3D space shapes. And the representation in X, Y, and Z coordinates is literally the same that we do for pictures. So in pictures, we have a red, green, and blue channel, and then an associated pixel to all of that. So it's a three-dimensional matrix. And without doing anything, we can represent a protein as well in that form, so X, Y, and Z channels. And then we don't really need to do anything to the AI architectures. Like we can use diffusion models like to generate pictures like DALL-E. Without changing that, we can train that on protein structures, and then we have a model that directly generates protein structures as well. This is why it has been so successful. Biologists didn't have to do any changes to the NLP architectures to start producing new sequences that work very well. And we tend to forget that proteins are dynamic, but we are seeing a lot of advances in the field of video generation that sometimes gets even a little bit scary. But with deepfakes, we are seeing what is possible these days. And I imagine a lot of advances into trying to model protein dynamics in the next months, maybe, or years to come. Without maybe going here, so we focus on language processing because at the end of the day, the function and the structure that a protein is going to carry is all encoded into the amino acid sequence. So in my view, we can, of course, train models that produce a 3D structure, but I think that whenever we have such a rich language and so many protein sequences that we can also take the architectures used in NLP and train directly on protein sequences. But before delving into how we do that, maybe you don't know very well the architectures that are behind the current language models. The models like ChatGPT are based on the transformer architecture, which was envisioned by Google in 2017, so almost now 10 years ago. And originally they developed it for translation, so they wanted to improve Google Translate, which maybe you remember, but at the time was very bad. So they wanted to get a model first for going from English to French, and they did also English to German. And in very short, so very high level, this module had two, architecture had two modules, an encoder that is capable of taking as input text, then through a mechanism called self-attention, it's able to pass that information to a decoder that it produces an output. And this could be the translation, or it could also be answer or summarization. So any text-to-text process can be modeled with a transformer architecture like this. But then other researchers started looking at the modules separately, and OpenAI took the decoder alone. So this is what we have today for language models. We have a module which is called the decoder that is capable of learning the probability distribution of a given sentence by splitting it into tokens. So what the model does is, given all the tokens that are in the sequence, it predicts what is going to be the next token. And this way, we can learn the probability distribution of tokens. So we have like a big box of tokens and what are the probability of all of them. This is very powerful for generation because then as input we can give the model a few tokens and then it's going to continue the sentence one after the other because it knows the probabilities of the previous ones. This is exactly the same that we do with proteins because we have a sequence of proteins and then the model can learn the probability of the amino acids. So given these 5 amino acids, what's the 6th and so on. It goes by steps. And this has been very powerful for protein design because we have lots of sequences, like people all over the world having collecting DNA samples from metagenomic efforts and also sequencing mammals and so on. So we have these big, large databases of DNA that we can then translate to proteins, and we have millions and millions and now billions of protein sequences that we can train the models on. The way this works more technically is that for the first model that I trained, for example, which I call ProGPT2, we take a protein database of sequences, like, for example, UniRep50, which at the time contained only 50 million sequences, and then we train that with this architecture on graphics processing units, so GPUs. At the time, I was very lucky to have access to over 128 that I trained the model in over 4 days. And this gave me a model with over 700 million parameters that once it's trained, it's able to speak. And this speaks very fast. Like it can produce many sequences, which is a blessing, but also a curse. Like we don't know how to analyze them often. So in English, we will just look at the text and say, oh, okay, this kind of makes sense. It's grammatically correct and his orthography is fine and so on and so forth. But here we don't speak protein, so we don't know if this is meaningful or not. So we usually have to go to the lab and start experimental characterizations. One thing what we can do first before doing that is to analyze in silico whether the sequences make sense and where they are. So the very first thing I did when I trained ProGPT2 was to look at the generated sequences, which are the ones that you see here in white. So these are the predictions of the structures, and I wanted to see where in the landscape of the protein space they will lie. So you see here, these are natural proteins, so sequences of proteins that people have been characterizing from different species, and then I put them in different colors depending on into what class they belong to. So whether topologically they are like alpha barrels or beta barrels or like the different specific 3D topologies. And then what I observe is that most of the new proteins kind of like are around a specific given previous faults. So like they tend to be, so the model tends to explore regions that are new but around previous clusters. But also from time to time it generates things that are completely novel. Like this sequence is only 9% identical to any other protein on Earth. So it's very good to explore unseen regions of the protein space. Given that this work, we've been training many models over the last years. I will focus today on the second one because ProGPT2, as I said, is able to create new proteins in anywhere in the protein space. But sometimes we just may want to have a solution for a specific application. Like we may want to specifically design carbonic anhydrases and not simply sample random sequences in the world. So this is when we train sign control, which specifically generates protein sequences for a given user-defined label. For sign control, we've been testing this experimentally many, many times. I will focus on a few systems. There we are. So we test every time we do an experimental characterization. This takes some considerable effort and time, as long as economical resources. So we've been over the years collecting data that this seems to work very well. And we are very happy that we can create new proteins that are functional in remote regions of the protein space. And I will focus on a few of these systems. The way the model was trained without going into a lot of technical details was by taking a protein database that is called Brenda, that contains enzyme sequences that have been annotated. So for every sequence, we know what catalytic, for every enzyme sequence, we know what catalytic reaction it performs. And we seem to have, like, we humans, a lot of favoritism for some easy labels. It seems like, for example, for this catalytic function, we have over 1 million sequences, but for some others, we have maybe 1 member. So this is a very unbalanced data set. What I did to try to solve that is to pass the model the information of the catalytic reaction, which is categorized with these 4 letters, so the numbers, and then the model is able to infer that whenever there are 2 sequences with the first number being the same, then it means that overall the model, that those sequences are catalyzing the same overall reaction, like a hydrolysis or an oxidation. The more we go to the right, then it means the model has some more specificity on the chemical reaction, like it could be, oh, these are 2 oxidations for sugars, but this one is doing glucose and this one is doing galactose. So the model learned that, it was able to transfer information from all the labels. And we apply this to several enzymes. For example, the lactate dehydrogenases. The lactate dehydrogenases are, for example, in our muscles, so they go from pyruvate to lactate. And then what we did is to generate from the model several of those, and we tested them in the lab, and we were happy to see that many of them are more active than those that we have in nature. And then Ramiro took a few of them, and then he characterized them, and then along with Silvia, they took them and then made them powder, so they were able to put them into cascades. So what we do is to introduce the enzymatic, the new designs in here, and we're able to go all the way from here to there, and we have some preliminary conversion. So we were very happy to see that just by pressing a button, we get some new sequences, that new lactate dehydrogenases that already work out of the box without any optimization in cascades that are fully synthetic. We also designed some alpha amylases, and this is very important because we had always, up to this point, before we did the alpha amylases, we had always designed 8 to 10 maybe enzymes at a time, so it was very hard to collect the statistics. But this time we did 200, and we were able to see some statistics of like what are the activities that the sequences display. And then we saw that many were better than the wild type, which is like the baseline, so like one of the original sequences on Earth. But then when I played a closer look, I saw that, yes, well, the generated sequences are better than the wild type, but also because in the training set, so what the model had originally seen, there were also many sequences that were better than the wild type. So it was a good kind of moment of, well, the models work and they produce new sequences that work well, that are new, that are active. However, it's because they are sampling, they are, they learn their training set and they are sampling the similar properties that they have. So I had a look at other properties, ignore maybe the properties themselves, but what is important to look at is, so the training set had the properties is always shown in gray, and then the generated sequences are always shown in blue. So you see that for whatever property, the generated sequences, so the model produced something that resembles very well the training set. And this happens for these 3 properties, but also any other property. So we have to be very careful to what fit into the training set, and it opens many philosophical questions of how do we improve the generation process. It may be that we don't want to specifically this level, this range of properties, but we may want to be better. How do we do that? And this is indeed a classical protein engineering problem. In protein engineering, we usually have a given property, which could be activity or the stability of the protein, and we may want to start from here, and traditionally people were doing little steps, so little changes in the sequence of amino acids so that we can go step by step up to the top of this local mountain. And this worked very well. It's usually often called directed evolution. But what if we want to go there? What do we do? And especially with protein language models. Because as we've seen, protein language models, they are good. They sample very long spaces, but they are just not the best sequences that you could possibly have. They are just as good as the sequences that you had in the training set. So what we thought that we could do is to apply some reinforcement learning paradigm. What we can do is, well, we have the first generation of proteins from the model, then we test them in the lab, and then we can tell the model which ones were working fine and which ones were not. And with this information, the model can go improving over time. So we see that ideally we can go from the valley of this landscape up to the top of to the maximum on the highest mountain. And this is what we call also reinforcement learning and agentic workflows. And this seems to be working very well as well for large language models. So whenever we see these days anything on ChatGPT, often you are asked something like, provide feedback, or how useful is this model? Then you are providing literally feedback which goes to the model and improves over time. We are doing something very similar here. What we do, for example, is we have a model like sign control, and then we specify a label like, oh, give me a carbonic anhydrase. Then the model generates some sequences. We test them in the lab. Some of them will work well, some others will fail, and all this information goes back to the model. We do this over runs, and this seems to be working well in silico and in vitro for a few of the first tests that we've done. For example, in silico, for the specific case of carbonic anhydrases, they mostly they have very different topologies because it's an important functionality that many different organisms, so that evolution has converged to have in different topologies. But often, if we ask any model to generate carbonic anhydrases, it will give you a bigger proportion of beta to alpha. So it will give you more often this shape than this another one. But what if we want this topology in here? How do we move the model how do we guide it to go from here to there? And what we can do is to, punish whenever it generates a beta carbonic anhydrase, but reward it whenever it produces an alpha carbonic anhydrase. And this is what we did. We start from here, and then we go rewarding and punishing whenever it does the right and wrong generation. And after 5 iterations, we were able to have mostly alpha carbonic anhydrase sequences. So we went from here to there. And we wonder whether this will be some sort of exploration trade-off. So maybe the model, is generating more alpha carbonic anhydrases, but they are all the same. But in fact, they are not. So when we look at the sequence diversity among the generated sequences, they tend to be very diverse. So we have many clusters at 90%. So it's able to go from here to there without compromising the sequence diversity. Then we went also experimentally to the lab. We've done many trials for this reinforcement learning paradigm, but I'm showing you the design of EGFR binders. So EGFR is a receptor that we have in cells, and is often involved in certain cancers. And it's been like a long-standing goal in therapeutics to design better binders, better inhibitors for this receptor. So we designed a few receptors with the model, the first round, and a few of them worked with this affinity. And then we were able to obtain this very first round. So we gave this information to the model, and this produced, the model learned from this first iteration, and it produced a second iteration, which is what we see here. These are the first iteration of the model. So these values are the lower, the better. This is a dissociation constant. So the lower, it will mean, the tighter is the binding. And it will take stronger forces to pull it out if it makes sense. So it will be binding for longer. And then we reinforce the model with this first iteration, and then we produce this second one, which has a few data points in here that are binding matches strongly to the receptor. And then these 2 rounds were given back to the model, and then we produce a third round. And this is what we see here. So we see that the model in the third round produced binders that are binding more strongly to the receptor. When we look at this, we wanted to go for a 4th round, but we already saw that I didn't plot it here for this figure, but patented and wild-type receptors, binders are not binding as strongly. So we believe we reached the limit of what is physically possible for these peptides to be binding as strongly. So I think it's also a good time to stop. One very interesting thing that we can do with this sort of reward models is that we can learn whenever a sequence is binding very well when it's doing very well in the task, what the specific contributions of each of the amino acids are. So we see that the reward can be decoupled into the different residues. So we see, these ones, the model this positioning here, the model is considering that is very important for binding, and which sometimes reveals, as we see in some interpretability studies, is not as straightforward for us humans. What we will think is driving most of the binding affinity according to the model in comparison to what the model tells us that is what the model thinks are the more important features for binding. So we are doing a lot of interpretability studies at the moment, which I won't delve into for the sake of time. But yeah, one very exciting thing is that we can couple of these to actual environments. So whenever we think of reinforcement learning, there is an agent, which in our case will be the model, and there is everything around it that is an environment. This has been grounded in psychological work. So whenever we as childs start learning, then we have ourselves, and then we are the agents, and then we go learning from the world, which is the entire environment. But we are lucky that in biology we can have automatic environments as well. So what we are doing at the moment is to couple the models to robots. So our idea is that the model ends up here, and then it can automatically generate the proteins that it's able to test, and then autonomously, our goal is that it retrieves this information, so which ones are the sequences working well, and then what are the sequences that are not doing so well, so that it can produce a second iteration and so on, all in a closed loop sort of agentic workflow. Our ideal goal in the end is that these agents are not only able to learn from sequences, but from any other information that we have in the world. We have a lot of annotations for many proteins, like the chemical reactions they catalyze, or the ligands they are binding to, or what are the global functions, or what organelle are they into. We also have sometimes dynamic information, and then we also have their 3D structural prediction. So we want to build that into bigger models, agents, that learn into these robotic platforms so that they operate autonomously and are able to produce and autonomously test the sequences. And all this, we want to couple to XAI, so interpretability studies of why the model thinks this is the best protein and not this other one, which is for us, biologists, not trivial. And yeah, without much delay, I want to thank my amazing team for all this work that we've been pushing for the last year. We've been here for a year now, and all the funding agencies for support. And I'm happy to take any questions. And again, thanks for having me here today.
Okay, time for questions.
Thank you for your nice talk. I had a question regarding the architecture that you use. You mentioned about a transformer architecture that you use the already existing, or you build the one in-house?
So for these two cases that I showed today, ProGPT-2 and Sign Control, we did not match. So it was a GPT-2 architecture that we tokenized differently and trained on a different data set than what it had been originally for GPT-2. For the later models, we are doing a lot of tweaking of the architecture as well. But for this one, was more like a proof of concept. Does it already out of the box work? And because we are lucky to have so much sequence data out there, it seems to. I imagine we'll need to do more elegant algorithmic choices as we run out of data for specific applications. This is what we see.
Thank you.
Yeah. Hey, thank you for the great talk and work. I have just a question wondering, like, how BTMs get into this work, like that the model can take into consideration what BTM that could happen to the protein that could affect the functions or the activities of this protein.
We haven't not worked on that, but there is a PTM language model out there. It was very recent out, Nature Methods. So they literally train the sequence model with the PTM information available in different data sets. This seems to work well for the prediction of PTM effects and so on. So downstream task of prediction and generation.
Amazing talk, thank you. I have a question regarding the interplay between the in silico training and the reinforcement learning part from the lab. Specifically, is the latency from real-world experiments that then provide feedback to the in silico training prohibitive, and if not, why?
We will think so, but it turns not to be the case. So once we have the assay deployed, it doesn't take too long. So we are saying at the moment, oh, well, we do the testing, and this the major delay comes from ordering the sequence to a third-party vendor. Then once they arrive, this takes like between 1 week and 2. The assay we can run it in-house in 2 to 3 days, depending on, so whenever we have it established, this is fast. And we are seeing that, or we will think that the PLM reinforcement learning is a lot faster, but as we are learning in the process, it takes for us even longer often the engineering part, because we need to tune a lot of hyperparameters and so on. So overall, I would say, to my surprise, that we are doing the experiments faster than the reinforcement learning part, but hopefully this will change one day.
Hey, thanks. Some of those sequences might work at like different pHs or different temperatures or experimental conditions. So if you're trying everything at the same, are you trying everything at the same experimental condition, or
Sadly, yes. And here we suspect many of those, because here we had worked previously with Basecamp, which is this company here in the UK, and they have metagenomic sequences from extreme places. So like they go to maybe places that are in the Antarctica and they do sequences, and then we fine-tune on their sequences for these enzymes in here. And then we tested a temperature of 45. So those that maybe resemble those in the training set that come from the Antarctica, I don't think they will be doing very well at the temperature 45, but we cannot really test at more temperature. So often what we try to think of is, well, what do we really want to optimize? And if it doesn't work at that temperature, that's it, but that's the temperature we want to optimize for. So we may be missing many false positives along the way. Yeah.
Thanks for the interesting talk. I was just curious, do you see any potential in vision transformers in this field? Because you drew the example with images, and transformer architectures actually can only take sequence data as input, so they're missing the spatial information.
Yes, they are having huge success. So I focus, if this is like the way I like to categorize protein design these days, we had physics-based design in the past, and now since 2020, we've been doing deep learning. People have been trying to either do inverse folding, so like to get a sequence for a scaffold, or this group of people, they focus on generated structures directly. And for that, what works best is to use either visual transformers or diffusion models directly, or flow matching, because with those you directly work with at the coordinate level. So a lot of people are doing this, yes, Rosetta fold diffusion. We focus on language because to me it was more trivial originally, thinking that the structure is already encoded here in the sequence, but there is a lot of work focusing on the sequence level, so they use visual or models that directly encode geometry, like diffusion and flow matching architectures. No, it's the same. It's the same input. So that's what makes the jump so easy, because in images you have a three-dimensional matrix, red, green, and blue. And for proteins, we have X, Y, and Z coordinates for every atom. So it's maybe more sparse, more like point clouds, but it's the same input. So no changes were made up for most of this work. And not in what we do, but the other groups. Yep.
Okay, I'd like to take the privilege as a chair to make a last question. For people like me who are very outside of all this, could you tell us like applications that you know of or that you envision happening of protein design?
That's an excellent question. I often wonder, I think the limit right now is more to the imagination of the researcher than the methods themselves. So sometimes I wonder, what do we do next? I think we are going to have a lot of advances in therapeutics. So people working on vaccines, but for example, basic things like the flu, these may work very soon. And I also think for protein engineering, so we had a lot of cases that we know of enzymes that work already well for certain processes, but they are just too slow to be brought up to industrial processes because it will be too expensive and pointless. But for those cases, I see the engineering is going to go a lot faster. So enzyme engineering, biocatalysis is going to be boosted by these processes now by a lot.
Wow, amazing. So let's thank again Noelia.
Thank you.